Hi class, let's start out with our next topic, bacteria and archaea. These are two of the three primary domains of life that have been categorized according to the WOS system. We have bacteria, archaea, and the one that you and I are probably the most interested in, eukaryotics. We'll focus on eukaryotes in the next chapter. This chapter, we're going to focus on these two domains. So let's talk about how they've been arranged. What are the structural characteristics of bacteria and archaea? They are definitely different from eukaryotes. We look at the way their DNA is packaged. Both bacteria and archaea do not have a nucleus, and they do not have histones. For those of you who do not remember, the nucleus is in the center of the cell. It's a double-membraned organelle that contains the DNA, and histones are small proteins that the DNA is wrapped very tightly around within the nucleus. You could think of a histone as a bobbin, and the DNA is the thread that is wrapped around the bobbin. If we look at the cell wall of bacteria and archaea, they have peptidoglycan and some other unique chemicals, but the big one is peptidoglycan, made of those NAG and NAM subunits. We also have internal structures inside of bacteria and archaea that are different from eukaryotes, particularly the lack of membrane-bound organelles. If the organelle is not membrane-bound, it can still be in our bacteria and archaea. For example, a ribosome. All bacterial cells will have a cell membrane, a cytoplasm, ribosomes, cytoskeletons, and one, or potentially more than one, chromosomes. Recall, a chromosome is a big piece of DNA with multiple genes on it. We'll cover that in more detail on our unit on genetics, but just to review briefly, a chromosome's a big piece of DNA with multiple genes within that big piece of DNA. Some of our bacterial cells will have a cell wall, depending on the species, and some bacterial cells will have a surface coating called a glycocalyx. We'll talk more about the glycocalyx later on in this presentation. A few, so some, but not all, bacterial cells will have flagella, pili, or fimbriae. These are extensions of the cell. An outer membrane, particularly if it's a gram-negative bacterium. A plasmid, which is a small circular loop of DNA within the cytoplasm. It's so small it's not considered a chromosome. Typically it only has a few genes on it. That's compared to a chromosome, which has many hundreds of genes on it. Inclusions, which are small invaginations of the cell membrane. An inclusion is like a mini organelle or a pseudo organelle. Some evolutionary theories believe that membrane-bound organelles started out as inclusions. Some bacterial cells, particularly the Bacillus and the Clostridium geni, will have endospore formation, and there'll sometimes be some intracellular membranes that will compartmentalize the inside of the bacterial or archaeal cell. So here's a generic bacterial cell that has everything. I kind of threw the book at it, so to speak. So if we look at this bacterial cell, we have a big fat flagella. Flagella are used for locomotion. I'll emphasize that again. Flagella are used for locomotion, not attachment. Over here, we have some fimbriae sticking out from the cell. These are very fine surfaces on the cell that sometimes are used to increase surface area or used for adhesion. We have, if we look at this modeled surface, we have a cell glycocalyx. The glycocalyx is fairly poorly defined. That glycocalyx is going to be kind of slimy and waxy, and depending on the species of bacterium, it will be larger or smaller. A pilus or pilus is a tube that attaches this one bacterium to another bacterium. You could think of a pilus as essentially a transfer tube that will form between bacteria that connects adjacent bacterium. We can have an inclusion. This inclusion granule is going to form an invagination of the cell membrane, and is typically going to have a lot of storage capabilities towards it. We have over here the nucleoid region, and this nucleoid region is where we have the big chromosome. We also are going to have these little dark blue dots representing ribosomes. And then finally, over here we have the circle of DNA. This circle of DNA represents a plasmid. And we can see also there's multiple layers here on this outside of this bacterium. It has a cytoplasm, it has an inner membrane, it has a cell wall, it has an outer membrane. So there's multiple layers that can form here to protect this bacterium. So let's talk about how we can categorize the bacterium based on their shape. Most bacteria are always going to function as an independent organism or a unicellular organism. Every once in a while, they'll team up and work together. And in those situations, they're going to be in a biofilm. Every now and again, there'll also be a nanowire that connects two or multiple bacterium that's used for communication. Nanowires are kind of the new kid on the block. They are very poorly understood right now, at least nanowires between bacterium for communication. So I don't have much to elaborate on with nanowires. I'll work on it. Our bacteria are, generally speaking, really, really small. They average inside of 1 micrometer or 1 micron. If we look at the different shapes, we have cocci. These are the round ones that look like little basketballs. They'll typically have a circumference or a cross-sectional length of, a outer diameter of 1 micron. The rod bacterium, or also known as Bacilli bacterium, are typically going to be 2 microns long and 1 micron wide. Depending on the bacterial species, there's going to be a little bit of variation in the shape and the size of our bacteriums. This variation within a species is known as pleomorphism, morph meaning shape. So we can have multiple or slightly different shapes within a single bacterial species. So here we can see some spherical bacterium. These are the cocci. We have the bacilli. They are elongated rod-shaped bacterium. And here we can see on the right we have some scanning electron micrographs of our bacterium. So here's that cocci, nice and round. Here we have our bacilli bacterium. If the bacterium's kind of squiggly, it bends a little bit. So we can see there's a little squiggle right here. Those are known as vibrio. If the bacterium is very loops back and forth on itself, we'll say 3 or 4 times, it's known as a spirillum shape. And if that bacterium loops back on its forth on itself about a dozen times, or more than a dozen times, it's known as a spiret. The difference between a spirillum and a spiret is with the spirillum, they are smaller and don't loop back and forth on themselves as often, whereas with the spiret, they loop back and forth on themselves very often. The thing that causes the spiret to loop back and forth on itself so much is it actually has a periplasmic flagellum, a flagellum that's sandwiched in between the cell membrane and the cell wall, and that's what gives it its squiggly appearance. And then every now and again, you'll see some bacterium that have branching filaments coming off of them. These branching filaments are usually going to be associated with fungal infections, but every now and again, you'll see them in bacterial cells. We can also categorize our bacterial cells based off of how they interact with each other. So here we have coccus, bacillus, vibrio, spirillum, spiret, and branching for the shape of the cell. And we also have some terminology for how the cells are connected to each other. Now, keep in mind, the bacterium are typically unicellular organisms. They can all exist individually. But as they start to reproduce, they preferentially will link together in specific ways. If we have just one bacterium, all by itself, we don't use a special name for that shape. It's just nothing. But if we have 2 bacterium stuck together, we call it diplo. And since these are round bacterium, I'm calling it Diplococcus. Diplo for 2, coccus for spherical. If I have a chain of bacterium, the prefix that we use is strept. And since they are spherical in nature, we use cocci, so streptococci is a chain of round bacterium. If my bacterium is a group of 4 bacterium, I call it a tetrad. And if it's a 3-dimensional group of 8, or a cube of cells, it's called a sarcina. So we can have a tetracoccal and a sarcinococcal. And then finally, if we just have a giant bunch of bacterium, kind of all mishmash with no rhyme or reason to its shape, we call that prefix staph. So here we have a big random clump of round bacterium, or spherical bacterium. We will call that a staphylococcal. So to reiterate the figure that I just talked about, we could have a single bacterium, we could have a diplo, or paired, tetrad, for a group of 4, staphylo for an irregular cluster, or strept as a prefix for a chain. So if we look at bacilli, our rod-shaped bacterium, we could have, again, those bacterium, just like with the coxal bacterium. With the bacillobacterium, we'd have a diplobacilli, we'd have a streptobacilli. Something that's unique for the bacilli bacterium is we could have a palisade. A palisade is a chain of partially attached with a small hinge region at the end. And this is named after an architectural feature. The spirella bacterium are going to occasionally be found in short chains. Generally speaking, the squiggly nature or the odd shape of the spirella bacterium makes it difficult for them to attach to each other. And then the spiret bacterium is very, very rarely going to remain attached to each other after cell division, because each spiret bacterium is going to be constantly moving and flexing itself as its internal flagella starts to spin and wind up and perform locomotive functions. So, concept check. Let's pause for a moment here. Which of the following structures are possessed by some, but not all, prokaryotes? So when we use this term prokaryote, I mean bacteria and archaea combined. This is an old school term. So you can pause the video right now and flip back to your PowerPoint, or you can rewind to go back to the beginning of this presentation. So, ready, set, go. Get me an answer. 1, 2, 3, 4, 5. Okay, so hopefully you've done that at this point. Let's go over the answer right now as a class. What is going to be in some but not all prokaryotes? I'm going to rewind here. All prokaryotes have a cell membrane, cytoplasm, ribosome, cytoskeleton, and a couple chunks of DNA. Some, or most, will have a cell wall or a glycocalyx, and some, but not all, will have a flagella, outer membrane, plasmid inclusion, endospore, intracellular membrane. So I go back to the question. The correct answer is D, flagella. Some, but not all, prokaryotes will have a flagella, but all prokaryotes will have a ribosome, a cell membrane, and a chromosome. If you have any questions, please feel free to shoot me an email or post them in the discussion board. Happy studies!